Got it. Got it. Makes sense. Okay, thank you.
So yeah, this is Liponema. Sponge are characterized by these very short tentacles. Characteristic.
Looks like we have a...
What? Oh,
I was just here comparing with our navigator, so about our position. So we'll try to make it here to the top of the ridge, and then going to turn northward. Northeastward, and follow the remainder of the dive on this top feature of the seamount.
Coral on your skid.
Let's go on this guy. He looked a little different.
Beauty.
Yeah, and I think that's...
So co-pilot, I think I'll give him the same thing again.
025.
I believe that's at Neoscepolis, yeah, and Bruce agrees. As Andre had identified a little bit earlier in the dive. Beautiful. Absolutely spectacular imagery.
We still got another couple minutes.
Yeah, this is good. The long pectoral fins are an identifying character. I have to look up which one, but it may be macrocheir. And a little bit of that video showed the photophores on the ventral part of the body, and that's another thing we can use.
Yeah, thank you, Bruce.
I'm gonna have to leave him soon, so go ahead, video.
I think I just gave a species name that may be wrong. There's Neoscepolis macrolepidotus, and that looks like it could be that species.
Pushing ahead, Dan.
All right. It's pretty flat, so it wasn't too alarmed.
Yeah, it looks like we have quite a few fish here on these pile.
All right. What do you think about taking over?
Sure, whenever you're ready.
All right.
A-okay.
So we're just going through a quick personnel change here, the ROV team.
Okay.
Current depth here is 724 meters, and yeah, the oxygen levels continue to rise. We're above 2.1 now.
Bridge nav. Yeah, same move. 40 meters, 025 degrees, 0.2 knots.
Dan, it may be an illusion, but I'm seeing lots of white dots inside those corals. Maybe
Good copy. Thank you.
gastropods?
Inside which corals?
Just seeing,
Oh, just in general?
in general, circular dots.
Okay.
They seem to be patterns.
Okay, we'll try to get a zoom if we see one.
There's a white thing on the right edge of the screen.
You want to take a look at it?
So we started to dive about 7 hours ago, and at that point we landed about 650 meters shallower, deeper than what we are right now, and in the early parts of the dive collected a geological sample, and that was the first sample ever collected from this seamount. This is actually the very first survey of this seamount.
Yeah, that current
and then worked our way up the slope, and just about an hour ago collected a very unusual bamboo coral that's
Be good practice.
very different from the many species of bamboo corals that we have here in Hawaii.
Zoom in.
Camera
Quite
is
distinctive.
80 degrees.
And interestingly enough, after collecting that, we did see a second species, a second individual of what appeared to be the exact same species that also had these unusual structures on their polyps. We have about an hour and a half remaining on this dive.
Quick zoom rolling, and then we'll have to pick up and go.
Okay.
Push out ahead of
Dan,
it.
can we get a look at this yellow coral if you haven't done so already?
It's on the screen right now, is it?
Oh, yeah, sorry. It's just the video
Oh,
is choppy and delayed.
Yes.
See it now.
Okay. Video
So yeah, we'd
I
seen
was
a...
wondering if it might be,
Thanks, Roland.
sorry, I was wondering if it might be an Acanella just
Right.
because it appears to
Pick up
be
and
growing
push
in
off.
sediment, and that's a very common feature of Acanellas, but up close it looks like that's not the case, probably just a Chrysogorgia again.
Yeah, so that's definitely a Chrysogorgia, and we'd seen something earlier, and might be a young
Camera is
Chrysogorgia
all the way
stellata. It has a kind of this roundish overall colony morphology.
Straight ahead.
But yeah, very sparse, the population of corals here. Substrate is, of course, a lot more rubbly and not as
Fish. Looks like the one we got already.
not as stable as the hard substrate we had earlier on.
There he is.
And so yeah, that's another thing is we're looking for, of course, places where we think we find dependable currents, because that's one of the predictors of where you might find corals. But it's also the substrate, and having stable substrate, hard substrate, and so it's an intricate interplay between different factors that determines whether a habitat is favorable. One thing that I think is quite informative from this dive in particular is that we went straight through the oxygen minimum layer, and we did not see a concomitant change in the abundance of corals. So there was definitely a different group of corals that was abundant in that group, but they did not disappear or really
Okay.
decrease substantially in abundance, which is one of the
These are
things that we
somewhat
were
similar
particularly interested in. Is these oxygen minimum layers are considered to be or a general trend of a lower abundance of animals, but I pointed out that several studies throughout the world have actually shown that there's some species that are become more abundant towards the edges of oxygen minimum layers.
Okay, let's go in video. Just get a quick view of it.
And so this is another sea pen, so that's the genus Penachula and Inflata for that inflated kind of morphology. Looks like it's puffing its chest. And yeah, very sparse the amount
I
of
think they're good with it.
animals we see among this debris
It's
of
more
coral
just for
rubble.
practice for me.
A couple
God,
of
there's an enemy above it.
sea
we haven't
pens we've seen, and
Yeah.
some anemones, and a couple of these chrysogorgid corals, but not really prime habitat for corals.
Where do you see it rolling?
You've turned away from it.
Oh,
It's already passed.
okay. Well, next time you see one, let me know.
Yep. They are a little orange.
And
We don't know if there was ever any done out here.
You've seen
any
that
what?
guy.
Sorry, go ahead.
Can you take a look at it on the scale
I
if you'd
say,
like?
I guess we don't know if there was ever
Do you
any
have the
trawling done out here.
Get out ahead of it.
Not
Yep.
that I'm aware of. Oh, that's a
Bunch
nice
of that.
aggregation of
It's
Brisingid.
a little
forced
tilted
up.
up.
It's a bit deep.
Strange.
Bruce might have a little bit. So Salmon Bank, that is actually to our about 20 miles east of us, that seamount has a flat top that gets to within 60 meters of the surface, and there's quite a bit of fishing that lobster fishery did work there, but yeah, this is a bit a bit deep for the bottom fishery. But I don't know, Bruce, do you have any about that?
Okay, we're coming up on the end of the move. I'll give it
Yeah, I'll
one more
chime in. There
40,
was
and I think
one
we'll
of those
have vehicles
unusual eels
very
to
close
the
the
left
waypoint.
under that ledge, by the way, one of the sorcerer's eels. But
025,
there
okay.
has been trawling up in this area, a little
Bridge
north
nav.
of here, not quite this far south, for some large aggregations
Yeah,
of
we'll do one more
pelagic
move,
armorhead,
40 meters,
Amlepenthes
025 degrees,
and alphonsins,
3 knots.
Barrack splendens, and Barrack decadactylus. I don't think the trawling occurred this far south. It did occur at the Hancock Seamounts to the north of Curie
Good copy,
Atoll.
thank you.
And some of the banks, one of the ones I was interested in looking at
Two fish over the
earlier.
ledge.
I see that eel up there on top right, by the
Shot
way.
again.
You're fine. My camera's at
Anyway,
40.
Oh, okay.
so trawling has, as Dan
Looking
said,
off of
it
it
looks like
to
there's
see
two
if
of
I
those
can find
eels.
stuff for you.
Anyway,
This is
trawling
good.
up has
Can't
occurred
tell. Some either another seep in or
in the region a
Yeah,
little
snaps in.
further to the north, but I don't think
another
it's
seep
occurred
in.
here.
Eels sticking
It
out
looked like
of the
Nettis stomatids
tentacles
to
or
me,
something,
Bruce.
Oh, yeah.
reaching out from
Yeah.
the
They look like the
crevice. I got really
Benefica
excited for
again,
a
but
second.
they're two
so
of them
to the
together.
left. is
Yeah,
a
maybe we
bunch
could
more
take
of
a
those
quick look at
percentages.
those eels.
You want to take a quick look at the eels, Watchley?
Yes, please.
Okay.
Also
Camera
looks like a
is
black
90
anemone.
down.
Okay, well, let's just do a quick zoom. Do the best I can for now.
You have some
Bruce,
problems
this looks
flying.
more like parviceps to me, doesn't it? Netistoma parviceps?
Yeah, I agree with that. So
sorry,
a different
Roland.
genus than what we saw last year, and one that we've seen from the
Trying to
Hurl
feel for the
dive. So that's
current.
why I wanted to take a look. So good, great.
Excellent.
And you know what? That the closer we zoom in, the more lithified that calcium carbonate substrate looks.
Okay, sorry, Roland.
So we go with
Doing
it.
well.
Watch lead nav.
Go ahead, Nav.
Yeah, we just started a last 40-meter move, which should bring us in
Center
about
on his head, I
5
can
minutes
snap
right
on it.
to the
Otherwise we
waypoint
can move
2.
on.
The
Let's
camera
move on.
is all the way back.
Yeah, let's move on.
I assume we'll pause there and then head off to the northeast.
Copy that. Yeah, that's exactly what we
If
want.
rotate
Thank you.
up and left, there's about 20 of those percentages. That's a
Group of
second
them
group.
So yeah, interesting to see these aggregations of the Brisingid sea stars. Bruce, one more question there. You talked about this armorhead fishery. Do you know what kind of depth ranges they were targeting?
Oh, that's a good
Kinda
point.
leading
That
on
was
purpose.
about half the depth
Camera's
that we're at
55.
now. So
Okay.
they were looking at around
Yeah,
250
I'll get out ahead
meters
here.
or so
Yeah,
for the
that's fine,
seamounts to the
catch
north
up.
of here.
You're
So
fine.
this would be a little deep for the trawl fisheries that were conducted in the region. And that's a very good point.
Yeah. So yeah, that was my first impression. Yeah, because this is quite deep. Yeah, the top just gets just below 700. I think 660 meters is the very top of the seamount.
Coming
You know,
back
although
down
the
for
armor
you.
had go down to I think 800 meters, or I may be not quite right about that. At least the alphonsin get that deep. So, but we aren't seeing any of those types of fish here in this dive.
Yeah. So yeah, just to give everyone a heads up, so we are quite close here. That first waypoint on top of this
Yeah,
this kind of flat plateau. So we should be reaching that in about 5 minutes. We're only 40 meters away from it, and at that point we will head towards the northwest. And there's kind of this flat plateau, so the terrain should flatten out a little bit more. Yeah, we just passed 700 meters. And yeah, should be able to spend our last hour and a half on the top of this flat area, kind of in the shallowest portion of the seamount.
Okay, zoom in video.
And so yeah, these are different sea stars, as Andrea just pointed out here in the chat room. So this is in a different genus than what we'd seen previously. These are in the genus Novadonia. So yeah, we're starting to see different species that we did not see on the deeper portion of the dive.
Tighter. Try to hold it steady. Okay, that'll do. Thanks, Roland. Actually, is there anything in particular we're keeping an eye out here for, whether geological or biological, or
No, I think
just doing a survey?
I mean, at one point there might be a opportunity to collect a second geological sample, but yeah, if we can make it to that top and then.
Okay.
So yeah, a lot of this more yellow substrate appears to be just fossilized carbonate reef terrace type of structure, not unconsolidated sediment. Kind of interesting type of.
So Jonathan, would you interpret this carbonate as a deeper carbonate formation, or was this kind of a touch and go at the surface because the summit of the seamount really doesn't have a flattened top like a feature that was above the sea level or right on it?
Oh man, we're so out of my my specialty in offering any kind of like knowledgeable comment, but yeah, you're right. I mean, it's an obvious observation of the morphology of the seamount. Does not have that atoll or flat-topped morphology to it. So obviously it wasn't at the surface for a very long time.
Even though we're pretty much right
But
away
something
from it.
had to be able to precipitate these more lithified carbonate facies. So presumably this summit of the seamount was at least a near-surface environment at one point in its history.
Yeah, let's take a look at this urchin.
Okay, we'll go for the urchin.
So we've done the move.
Okay. Probably keep it moving.
Okay. Should I go more than 40? It's pretty level.
Yeah, sure. Okay, zoom in, roll. Just kicked up some sediment.
So guys,
There's a forest fire there.
it's like it's an Okeanos. I think we have one
Sorry for the
feature
sediment.
only of genus in this family in Hawaii. So this would make the second observation
I can reset up rolling on the other side of the
type of urchin here.
something I didn't think about was the current.
Thank you, Chris. You broke up there. You mind just typing in that in the chat room? Yeah, we barely could hear you.
Okay, we're going to dogleg off more easterly. I'd like to do a move 70 meters, bearing 080 degrees, decimal 2 knots.
Just the ID, Chris.
Good copy, thank you.
Thanks,
Let's try
Chris.
this again, rolling.
Tilt the camera down a little bit.
So Copilot, we're going to go off about 080.
Copy 080.
Yeah, beautiful close-up view of this urchin and genus Okeanos. And thanks, Chris, for that ID.
actually, I'm looking at the other picture, which is in the Hurl Gallery, and the best we could do was Echinidae, and thought it could be in that family, but I think it's the same urchin. And again, it's the second time it's ever been imaged out here.
Yeah, very neat. Yeah, I think throughout the dive we've already imaged a few things that are unusual, and including that one bamboo coral that we also have a specimen in our sample box. And
now we're going up on
I know several people are going to be very, very excited about analyzing those specimens.
We're not going to get to it.
All right. Well, we can try.
Give you another hour at least.
We'll get as far as we can.
An additional hour.
Well, we can try.
Let's speed up.
Flat enough.
I mean, we're going 0.2.
Yeah.
Is 0.3 reasonable on this terrain body?
And, yeah, so
I think so, as long as we don't want to
as
stop
soon
it.
as this terrain started flattening out,
there's
that was
real
around
Gold to get there, we can.
750 meters or so. We
can check in with Watchley and see if he'd rather stop for close-ups or make ground.
Right.
I think the consensus is, if we see something unusual, we'll image it. And then we'll make it to that waypoint 1, and then just as long as we have bottom time remaining, we'll just cruise on that top feature.
The way we've been doing.
Exactly. Yeah.
We just got to the waypoint.
Okay, perfect. I'm happy. Yeah, that's great.
Now we're headed east at the same speed, but we won't make the main peak.
Yeah, that was understood. That was an ambitious goal, but
well, we could if you wanted to speed up, but then you wouldn't have the opportunity to check stuff out as freely.
Yeah, because it looks like the actual true summit of the seamount is those next close contours to our east.
That's right.
Correct.
So is that waypoint 3? That yeah. So that green.
Beyond that.
Oh, okay.
Is here. At this point.
Yeah.
Looks like the real summit.
Yeah.
Is whoops.
I think if we cover some ground and shoot for that summit, that would be kind of a neat end to the dive.
I agree. Yeah.
Yeah. Let's bump it up to 0.3 then.
I don't think we're going to get there even at 0.3. It's 450 meters.
Oh, goodness.
We've been doing about 150 meters an hour.
Something interestingly.
Yeah. No, I mean that was an ambitious goal. Yeah, we'll see what we can get here.
What was that, Roland?
Something interesting. Pink went off screen.
Ahead,
bottom left. You rotate left.
But there isn't that much left of elevation gain. So yeah, if I remember correctly, it was right around 660 meters was the very top portion, and we're now at 696 meters.
Just getting on the other side of the current.
So just another 6.
There's a little red ogocephalid on the bottom there. That'd be worth videoing.
Yeah, so that animal right there, bottom.
Yeah, absolutely.
So
let's zoom in.
Good.
So yeah, that's gonna be in the family Canasidae.
The ship's adjusting head 15 degrees or so to starboard.
Okay. Try to get him centered up for you, Roland.
Yeah, he's a little guy. Max in.
So, Bruce, I'm going to let you call out this one. It's the same one that we imaged in last year, correct?
Yeah, I think that's correct. I'll have to look at it in a little detail. I'm going to get the name of this wrong until I look it up, but it's something like Scolyssa squama or Solyssa squama erythrinus. The species about erythrinus is red. It's a species that Charles Gilbert described from the albatross collections back at the turn of the 19th to 20th century.
Yeah, take me a little more north.
My memory is beginning to fail. Like I said, it's something like Scolyssa squama or Solyssa squama. It was placed in the genus Dibranchus. which
Yeah,
was
we're
easier
gonna
to
wanna
say.
just,
And then Margaret Bradbury
move a
did a
bit
revision
further
and
in
put it in Eugene. It's impossible to pronounce.
050. Can you just
I believe
submit
it's
that change?
Solosquama erythrina.
Okay, I'll go with that. Great, thanks, Dan.
Sounds good. Thank you.
Okay, adjusting
Right.
to 050. Thank you, Dan.
Yeah, very. So yeah, as I was saying, so the top of this feature is only 30 meters shallower than we are, or thereabouts.
He's not moving, is he?
So
He's
yeah,
not scared. All right, let's zoom out.
Thank you.
Pulling out.
And your lasers are on for reference.
so yeah, there are a couple
Yeah.
comments here that this is in the family Okeanos.
About 10 centimeters.
A beautiful animal.
Okay, coming
Full
up.
wide.
Thank you.
Thank
Lasers
you,
on.
pilot.
Good spot rolling. All right, did we make a heading change?
And 050. I'm pointing there.
Copy that.
Take us a little more upslope.
Sure.
Also, and if you stay about this height, this angle of the camera, I can give you a further distant view without washing out. Very long
Starting to
eel.
see more of this
Straight ahead.
sea pens.
Oh,
Yeah,
yeah.
I see him, yeah.
Blends in pretty well. Good eye roll.
You want to zoom in real quick? I'll do my best.
Oh no, it's a coral for siphonophore.
Yeah, he's
Yeah, I don't think
A little
that's gonna
far.
swim away.
Do you want to go check it out?
yeah. Let's.
Okay, let's zoom out.
He will definitely blow in the wind.
Yeah. Try to get over this sea pen and then sit down.
I'm going to bring my heading around.
Okay.
Starting to move. He's undulating.
Yeah, that's
Oh,
a
that's cool.
siphonophore. That's very cool.
Just touch the ground. Try to keep with him.
Bring it around.
Yeah, good spot there, Dan.
That was rolling.
Oh, Roland, yeah.
Tilt down, I can zoom in.
Yep. Slowly tilting down as I pass around him.
You guys put the lasers on when you're done with the close-ups just to see how big this thing is?
Copy
Wanna zoom
that,
out,
Chris. Yeah.
Lasers are on.
He's
Yeah,
pretty
so
tall.
yeah, he is very tall.
I think you're going to be pulling me around, so I'm going to lose
Okay.
you for a second just to give you some more tether.
It's interesting as we look at this to consider that
Ready
this is
for
also
zoom in?
an idarian and it's also
Pretty
colonial,
dark
and so it's
background.
closely
Yeah, that's
related
nice.
to all these corals that we've been looking at, except that it's modeled to live in the water column. And the part that you're seeing now at the end, the right side of the screen, are basically, it's almost like a series of medusa, almost like a series of jellyfish that are all combined, and those are the jet
Got it for
propulsion
now.
for this
Be
entire
ready to see
colony.
You know.
And the parts that you see that are colored red and orange
that's
would be
nice.
where the feeding polyps are and the reproductive polyps. And you can see there's a long string of them, and they can
Pumps at the end are
possibly
pretty
break
cool, too.
apart. These would also have tentacles that could be stinging and so on.
Yeah, those are
Yeah.
Up, he's going up.
Yeah, that is a stunning imagery.
He's getting out of my light
That
pool.
is.
He's in your light pool, rather.
Okay. He's within it.
He's inside the vehicle.
Is with the
Slowly
lasers
back
on,
up.
on it. Possible to
Full wide.
Oh. Watch out for the thrusters.
He's starting to float away.
Yeah, there's a
I'm going to come up
It's
a
also probably time for
couple
you to
compliments
Sure.
here, and I
get back
absolutely echo that. Great job by our pilots and by our videographers to capture this magnificent image of this siphonophore.
I'll let him float off and we'll head out.
A very nice pirouette. Tilt down as you go.
So you
Okay.
Super nice flying.
Thank you, Roland. All right, which direction?
You can see
Head-
some of
make your
the
head
tentacles
0,
stringing off
4,
the
5.
lower part of the colony there, which
Oh, sorry.
relates
Yeah,
it
make
back
your head
to
1, 3, 5.
Cnidarian polyps.
135.
Yeah.
Okay.
Yeah. Yeah, so we were talking about that earlier, Scott, and there's a couple comments made. Yeah, so that's what
Ooh,
the
okay.
Cnidarians. So you have these
They're
stinging
serious.
cells that you'd mentioned, but you also have either a polyp and or a medusa life stage. So different groups have either alternating
So you have to keep
or
cutting over as
one
best
or
you
the
can.
other
Okay.
of these different life stages.
Up. My camera's coming down.
Okay.
You can see your light pool coming in.
Let's see it on the bubble cam. There we come.
Depth is 690 meters. Again, this is for
Alright.
those folks who joined us
9050.
later in the dive. This is a seamount.
an eye
It's
on
about 20 nautical,
Nice job getting
20
that.
miles west of Salmon
That's
Bank
good.
and about 20 miles south of the boundary of the Papahanaumoku Marine National Monument. And this is a seamount that we just mapped yesterday for the very first time.
At this point, but
Had never been surveyed, had never been
Still
sampled.
Looks like it goes off to my left, so
And
either
yeah, so these are the
060
first
or so,
images
I
of
think
this
we-
area and the
I don't
first
think they're gonna go very far that way. I think
real
maybe
data that we
side-
collect
Wanna
from this
snap zoom on
seamount.
this fish?
And so this is,
I'll try to center him up as I go by.
yeah,
I'll do
so
a-
he's,
I'll do a
he's
couple
hiding there in that
short ones
nice group of
I probably
Oh,
would
sorry.
just do
a little
asteroids.
fast.
And so those
Momentum.
asteroids or sea stars are all in the genus Novadonia.
East. That's going to take me off into the darkness.
Right. No,
Think
I'll,
we're
I'll,
good.
but
And,
I'll keep it short though, because I think
Yep.
if we go very far north, we're
Gotta
going
keep you out of
to
here.
And we
Before
have
we
on
go
the
fly.
periphery a lot of these sea pens, and those are in the genus Penachula, and species Penachula inflata. And yeah, this is a nice little cluster of life.
bridge, Nav.
And I don't know how
Yeah, we'd like to
Andrea
do
counted that so
30
quickly.
meters, bearing 045.
And she says there's 22
decimal
of them.
knots.
And yeah, that fish is not being very cooperative. He's hiding there.
You want it again, Dan?
Sure. And I'm setting my toe down here. Stand by
And
one
yeah, so
second.
Scott
Oh,
made
yes,
this
sorry.
a good observation here in the chat room that all these animals here, those sea pens and these sea stars, are all lined up here along the edge of this ridge. Where likely you have again accelerated currents, and those are kind of the features we've been targeting because we were looking for corals and sponges. But yeah, here the substrate doesn't be conducive for these large corals and sponges to settle and to grow. And so yeah, you have other smaller organisms that make use of these same kind of places where you have accelerated current flow. What?
Okay, and he's gone. Okay, did you change the direction? Sorry.
Yeah, we're going 045,
Okay,
but just 30 meters.
just get an idea of the slope.
Is that a sea star to your right? Sorry.
Saw a sea star earlier.
Which one?
Color.
Oh, down there.
Sorry, you don't have to look at it. I'm just.
It's a white one.
Looking for interesting things. Roland wants to see him too, so.
White one similar that we imaged earlier.
And a few people are commenting here on how.
Let's do a snap zoom. We'll just check it out real
Hey,
quick.
Dan. I thought I would chime in with all of this great Novadynia stuff happening.
Yeah, there's another one of your friends. There's I think Astenactus right there.
Also, immediately above that looked like a white
I think I'm still running a little behind.
dandelion.
Well, you're in for a
watching
treat.
video of a current running amongst the Novodynia.
Moving too much.
I guess the Astronactus is coming up.
They've seen that
Yeah.
multiple times today.
The Novodynias are definitely taking advantage of that current flow, and this is just a really wonderful. When I first saw them, I was just this has only been seen, I think, once or twice by Hurl since the original description. So it's just outstanding to see them again. And Novodynia is the same genus of persingid that is often seen in the Atlantic in the tropical Bahamian area.
Nice
So
job on that
it's
Symphony
interesting
4.
to
That
see
was
them.
pretty awesome.
Yeah,
Astronactus, of course, is one of
These
the.
are very
Oh,
colorful.
before I forget, Novodynia is also just kind of a curious lesson in taxonomy because it's the names are actually made based on Nordic gods. So the original name was
Back up again.
Odinia, named after Odin, and they found the name was already occupied by, I think it's a beetle. So the name actually means new Odin, new Odinia, Novodynia. Oh, here we go on the Astronactus. So, so just outstanding, this Siphonophore, and yes, the Astronactus looks like it's
All right.
many of the. It used to be something that I thought was really rarely seen, and now we're
Looking at my
seeing them
flybys. Rolling.
pretty frequently
Right there yet.
on these dives, which tells us a lot about sort of the relative rarity of these deep sea animals. I don't know how they survive because they're all, you never see
This
more
looks
than
like a top
one
feature here.
at a time.
We're just
So
going up on the
it's just kind of curious
Another 5
how
meters or so.
how they reproduce or any of that sort of thing,
Love to
or
have
if
your
they're
take
even
on
the
what
same
the direction
species.
of the ridge is. It
How
should be
far
about east.
is too far before there's another
D2
population?
Sam.
So anyway, really interesting questions, and I'm loving this sort of shallower area where we're seeing all of the other taxa. So anyway, keep up the good work,
Strike
and
off in that direction.
I appreciate the
We're
longer
way too
dive today.
serious
So
I would say
thanks a lot.
let's just cut the
Yeah,
corner.
and thanks for calling in, Chris.
Cut the corner. You can go
It'll be fine.
090.
Oh, it's,
No, so
it's
Oh,
getting
so
to be late
half
here,
I'll
but
move
zero,
I'm
70.
right down to the
070.
wire with this. So have a good night.
You too. Thanks, Chris. We have about an
075.
hour left on this dive, so hopefully we'll get you a few more images of. We've
Bridge nav.
kind of come to this more flatter part, if you will, of the top
Let's
of the
do
seamount.
30 meters, 30 meters,
uh,
bearing 075.
We started at close to 1,300 meters, the dive, and had a pretty steep slope, average about 45 degrees, and that took us all the way up to about 750. And
Copy, thank you.
at that point, the slope started to flatten out. We had slopes closer to 20 degrees or less. And as soon as it started to flatten out, we came across this very large pile of coral debris, which appeared to be at least multiple species from the good close-up images we got of some of this pile. And at that point, the corals and sponges that we'd seen before disappeared, and we just see isolated animals around these debris piles, and then a few of these clusters of aggregations of these sea stars
Copy that.
in the genus Novodynia. And then we've also seen a few anemones in between some of these patches, and several fish swimming through them, and some urchins, but definitely a very different community on this different substrate here.
Sediment.
So just slide and zoom in. We already got these guys, but just a little bit of practice while we're here.
I'll let you slowly rotate and I'll follow focus.
Yeah, so this is, yeah, you can even see there that there's strong currents. And those are kind of the things that I've been looking for before when we were looking for favorable environments for
Zoom out and I'll move over there.
corals and sponges. Now, here the substrate had just changed, and consequently we're also seeing a change in the community.
There's a either cup coral, small anemone to its right, right next to it.
Hey Daniel, can we get a quick zoom in on the red in the center?
Yeah, all right, coming right up. So yeah, that's a mushroom coral, so that's an Antimastus, looks like it. And at least two different species of anemones to its right. And yeah, these are typically found on hard substrate, and it's found as just a little patch to settle on there. And
Okay, trying to move to the anemone.
yeah, so we have at least two
Oh,
there.
I just lost one too.
There we go.
Did you want to see the anemone, Dan?
No, it's quite all right. Thanks, Dan.
All right.
And looks like we see quite a few of these clusters, aggregations of these sea stars here. Very neat. Yeah, so very few corals. That red was a mushroom coral, and here to our left appears to be another. Looks like a chrysogorgid coral from here, but very few corals here at the top. There's a sea pen to our right that we just exited the screen. Brownish.
They're changing pilots.
Yep, roger that. Yeah, so we're just going through a little personnel change.
Once you're ready, there's a sea pen off right that they wanted to see.
Copy.
Just as you guys were passing off.
Yeah, that looks like the last ship move just ended, so a little bit of swing. And then that should be it for now.
You're a little quiet there now.
How's this?
That's better.
Awesome. Yeah, so the last ship move just ended.
Okay. Well, let's catch the seat pen before we do anything else. You said to the right, co-pilot?
In this last I heard.
I can't hear watch leads.
They're muted right now, I believe.
Yeah.
Any of those.
Okay.
I am looking back at you.
Okay.
But the ship
Are you
just
still
stopped.
swinging, you think?
Yeah, but it's flat.
Okay. What about that one at top right, Roland?
I think it would be fine.
You're quiet too, because I'm not listening to you. There we go.
Not listening to me?
Not
That
on
can't
purpose.
be good.
Not purposefully. I'm not going to set down.
Copy.
Yeah, so we do see at least two different species of sea pens here,
Okay,
those
you wanna
large
start to come in,
inflated ones. And then there's, so this is Penachula, and to our right was the Anthoptylum.
Just hold there. Come wide, Roland. Let's get a better view of this guy. I didn't want to set down down current.
So yeah, Chris, I saw your comment. Do you think, I mean, I don't know how easy it will be to get. So there's a little bit of discussion. I might try to get one of these sea urchins here.
It's that white thing in the upper right.
I don't know.
That's an interesting white blob.
Okay, well let's
Yeah,
try
just
to.
suggesting it because it most likely would be a new record for Hawaii.
And so you mean that one that's kind of right of screen there on top of the rock, and I think I've seen several here right in front of us. Is that the one?
Yeah, that's it. Yeah, that's the one.
Okay.
It's that
Come
white
on,
one
let's
with
go.
sort of short spines. I don't know how, I can't give it how small it is or how easy it'd be to collect, but anyway, it's different.
Yeah, I mean, they look quite small. I don't know if they are very, very delicate or...
Science, we also have a sponge when we pull out we haven't seen in a few hours.
Yeah.
A little anemone to the bottom left there, or something.
Yep, or a tube-dwelling anemone, or Cerantherian. There is a sponge there in the background that, yeah, we haven't seen sponges for a long time.
Is that a Chris Kelly request?
No, I just pointed it out to them. Let them know that we've seen one and we haven't for a while.
Okay.
Not that we
I
have
was just,
yeah, so I think there's a couple of people who might be interested in as collecting possibly those urchins, and I don't know how difficult that would be. I mean, they look very small, and so yeah, that's a Farreid
let me try
sponge.
to turn for you, Roland.
Yeah, so no need to get really close. We know what it is.
Okay. So we want to try to collect one of these urchins here?
Yeah, how difficult do you think? I mean, it looks so small.
I think it's doable.
Doable, all right.
What do you think, co-pilot?
Is the ground hard or is it soft?
Right now I'm down on it.
Ground is fairly hard.
50, so
it does crush.
It's going to be tough, but I mean, you can take a swing at it.
Yeah. Well, let's come wide, Roland, if you're not already.
You're full wide.
Which what are they talking about?
These orange, I believe, right?
Nope.
Yeah, there's multiple ones there in view.
Pink balls.
What are we looking to sample?
There's also another rat tail off in the distance, kind of nestled in the little outcrop.
Yeah,
Watch lead, pilot.
Go ahead, pilot.
Looks like a
Hey, what exactly are we looking to?
But it looks like it might be a little hard to grab. It should have been a little nook there.
Chris, the one that's now kind of a little right of center? Is that all right?
Yeah, that one. It looks bigger than the other one, and that might be easier to grab. I'm pretty sure it's the same thing.
Yeah, I mean, we'll definitely image it, and I think that siphonophore is heading right for us.
There's a squid.
Oh.
Go
and
ahead,
a squid.
Go ahead at the squid.
No, never mind.
Yeah.
Okay, well, let's inch in here, see if we can...
How are you with your grip control?
What does that mean?
At the ability to stop the grip like halfway.
Pretty good.
Okay, because that's what this is all going to be about.
Okay.
You guys do understand?
Yeah, I agree with you, Chris. That's definitely the biggest one, and the other it looks like a bit more accessible than the ones that are in that crevice.
Hey, Sean, do we have any biosamples already?
I was told we do have one, but I don't see it written down.
So it's on the port side.
It's on the port side?
Yes.
Okay. Port inner or outer?
I believe outer.
Okay.
Hey, Daniel and Jonathan, if we can get a zoom of that fish, too, I think that's something different than what we've seen yet today.
You can zoom on fish. We're pretty much set up for that right now.
Copy that, yeah.
Is that Andrea?
Yeah.
Yeah, you want to go ahead and give me
And
some?
first crab.
Got hydraulics.
Oh, Bruce got it.
Okay, whenever you're clear video, I'm clear.
Yeah, Bruce wrote
So
in
where we
the
going? Forget this thing.
We can go starboard.
Okay.
Yeah, thanks, Andrea. Saw it, and thanks, Bruce, for.
I'm thinking maybe I'll try to just cradle it and not grab it. It's the pink.
Yeah, it's the pink. I mean, ideally, you just sort of you come around it.
Yeah,
and you
but
go
not
just enough to get underneath it,
Yeah.
and then pick it up like that. So you might not be able to jaw lock.
Right.
So think about where your controller is before you actually grab it. You want your
yeah,
HD
if you could
too?
set me up, yeah, that'd be perfect.
I'm gonna take your HD too.
That sounds good. Go ahead in on it. Let's do grip.
So yeah, if you just joined us in the last couple of minutes, we're just trying to set up for this urchin. That is most definitely a new record for Hawaii.
Oh, I need hydraulics.
Yeah, hydraulics are rare.
We're at a depth of 686 meters. We're on the very close to the summit of a seamount that was just mapped last night by the mapping team of this ship. We were not able to dive in the last couple of days because of rough weather, but we were still able to map and
take a second to get
covered a large area here west of Salmon Bank. There's a couple spots that showed up on the satellite data,
Do you want to?
and we drove over them and mapped three brand new seamounts
do you want to try to put it in
that
port?
I want to put in
were never been
whatever's
mapped
easier.
before, and the
this is a
seamount we're
sample
diving on today, a very
I'll put
tall
I'll put it
feature,
in
is
starboard.
Okay.
close to 13,000 feet
Yeah,
in
actually,
elevation.
I'm thinking now maybe I'll rotate and get that mid-screen.
And we're
What do you think?
rising from about 4,500 meters and then coming here to the surface close to 600 meters from the surface. That's the shallowest portion.
Helpful for me.
All right, video. Let's do a partial on Mini Zeus.
And yeah. In the earlier portion of the dive, we collected one geological sample, a rock sample that will hopefully give geologists a way to date this seamount. A little bit later in the dive, we collected a very interesting and unusual bamboo coral. At the very least, that's a new record for Hawaii. We have never seen anything like it, and looks like a few of our viewers that have done a lot of work elsewhere also have not seen that.
Zoom on HD1.
And now we are trying to collect this urchin.
Grip lock 1 doesn't even
That's
go
also
in.
a
Okay. Then you need something a little higher.
new record for the Hawaiian archipelago.
Grip force 1, sorry.
So this is going to be specimen 03 bio.
And that's 2. That doesn't want to go either.
Try like 4 or 5.
Yeah. Okay. Somebody left my... Okay.
Yeah, you got this.
Trying to get it in a way that I can see it.
starboard compartment nip. Just help you out a little bit. The mini Zeus isn't very useful.
Yeah.
While you guys are holding, can I show you a shot in your right screen?
What shot do you want to put there, Roland?
Just a quick multi-view. That's helpful.
No. Please change it back. Thank you. Oh, wait for that roll. Alright, well now my jaws don't want to close. Okay, there we go. Open. Wide on mini Zeus.
Fully open.
So now I can't grip lock, right?
Yeah, I don't know.
We can.
Oh, excellent. That's awesome. Alright, you got this then. Take your time. Oh. Oh, awesome. And you can put it in either side of that.
Yeah.
Doesn't matter. You don't have any more wrist left?
No, I do.
Oh, okay.
Oh, that's the roll there. Let's wait.
Let me know when you're clear.
I'm clear.
And we can all breathe. Outstanding job. So yeah, again, our pilots did a superb job.
Thanks, Josh.
Awesome job, man.
Yeah, outstanding job.
That was
You have some very happy scientists here on the chat. So yeah, that was that was a great job. A very
I think we're clear here.
delicate collection, and
all the way up.
I thought I brought it all the way up. Let me check. Yeah.
Okay.
And that one manip cam, it looked